Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.3, plasma membrane. Our goal today is to describe the role of all of the components of the cell membrane and how it helps maintain the internal environment of the cell. We'll be looking in depth at phospholipids, embedded proteins, as well as the overall fluid mosaic model of the cell membrane. And the suggested skill is Visual Representations 2A, describe characteristics of a biological concept, process, or model represented visually. So in taking a look at the structure of the cell membrane, we have to know what the primary component is and how it works. So this would be the phospholipid. Now, as you can probably tell from its name, it's composed of two sections, a phosphate group and a lipid. So it has a hydrophilic phosphate head. It's got the phosphate head right here is charged. It's attracted to water. It's going to want to go near the aqueous solution. Whereas there's also a hydrophobic fatty acid tail. And there are two fatty acid chains that make up this tail, and they could either be saturated or unsaturated fatty acid chains. So this one here is saturated, it doesn't have any bends, any kinks, while this one is unsaturated, there is a double bend. And this is going to matter when arranging all of these phospholipids together because it's going to change the distance between them. And within the cell membrane, they arrange themselves in a bilayer. There are two layers of them, which you can see down here. And why are they arranging themselves in a bilayer? Well, the reason is we've got hydrophilic heads that want to be near aqueous solutions, and we have hydrophobic tails that don't want to be near any water or fluid. And both inside the cell, intracellular, and outside of the cell, extracellular, we're going to have water. There's going to be aqueous solution. That's going to be what the hydrophilic phosphate heads are facing towards. So this could be the outside of the cell, and this could be the inside of the cell. But either way, and vice versa, but either way, the hydrophilic heads are going to be facing out towards that, while the fatty acid tails are going to be facing inside, away from the water. And that's why these phospholipids become arranged in a phospholipid bilayer. Now, the cell membrane doesn't only consist of phospholipids. There are also embedded proteins. There are steroids, namely cholesterol, which affects the fluidity. There are glycoproteins and glycolipids, and all of these can flow around the surface of the cell. Now, glycoproteins and glycolipids, we have to take a look at the name again. We obviously know what a protein is, but a glycoprotein is going to be a protein attached to a carbohydrate, while a glycolipid is going to be a lipid attached to a carbohydrate. And all of these are going to be embedded together within the cell membrane between our phospholipids. So if we look over here, this is what's known as the fluid mosaic model, because all of this is moving around, it's flowing, none of it is a rigid structure. But we can see we've got phospholipids, we have some glycoproteins and glycolipids as well sticking out. There are going to be proteins embedded into the membrane itself, and these could either go through the entire membrane or only through a part of it. The ones that go through the entire membrane are known as transmembrane or integral proteins, and ones only on the side are peripheral proteins. And these will assist in transport of items through the membrane, but we'll talk about that later. Now over here, we'll see on the inside, these are cholesterol. They're steroid molecules, they're cholesterol, and the amount of cholesterol can help control fluidity by changing the rigidity of the membrane depending on the temperature and pH. It can loosen up the membrane or it could make it stiffer. And glycoproteins and glycolipids, which we can see here hanging out the sides of the cell, these are going to be used for signaling and cell-to-cell recognition, because each cell is going to have its own set of glycoproteins and lipids that can tell other cells, hey, we're friendly, or this is our purpose. And the protein side chains of the glycoproteins can of course be polar or nonpolar, contributing to their reception. So here we have a picture in a little bit more detail of the fluid mosaic model. You can see it's a very expensive cell membrane, and it's primarily going to be, of course, phospholipids with phosphate heads going out into the environment aqueous solution outside and inside the cell, but there are also going to be glycolipids, glycoproteins sticking out of the cell for signaling. There are going to be proteins embedded into the cell or hanging out the side, which are going to be integral or peripheral, and there are even going to be cholesterol inside the cell inside the membrane affecting its rigidity. So this is how all of the components of the cellular membrane work together. This is the fluid mosaic model. So plasma membranes review, all you got to know are the structure and properties of the phospholipid bilayer and the fluid mosaic model. You got to know your phospholipids and how they have a head and a tail of different polarities. They're going to be arranged in the phospholipid bilayer, and there are going to be these other components embedded into this bilayer, namely proteins, glycoproteins, glycolipids, and cholesterol. So hopefully this was helpful, and next up we are going to be talking about membrane permeability and things that can travel through the membrane in and out of the cell.